Howdy, folks. Thanks for checking in to Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this second notes cast in our series on going beyond Mendel's laws of inheritance, we're going to be talking about sex linkage and the effects of in the environment on gene expression. Before we can talk about traits that are linked to the sex chromosomes, we have to talk about the classes of chromosomes themselves. So we have 23 pairs of chromosomes. The first 22 pairs in a karyotype like this are called autosomes or autosomal chromosomes. Sometimes we even call these body chromosomes. Not that they code only for body parts, but it's just convenient to call them body chromosomes to set them apart from the last pair of chromosomes, which are called the sex chromosomes. Now, it's not just because the genes for peepees and hoo-haws are encoded on these chromosomes. Those are spread out through all of the different chromosomes. But these are the ones that are associated with sex determination. Sex-linked traits, then, are those traits that are on the sex chromosomes themselves, not the autosomal ones, but that last pair of chromosomes. These were first discovered by T.H. Morgan working at Columbia University, working with fruit flies. Fruit flies are a good subject for genetics because they're really prolific. They make lots of babies, and they only take 2 weeks per generation. So you can go through several steps, several iterations, several generations in a pretty short period of time. Plus, they're pretty simple genetically. They have 4 chromosomes and 4 pairs of chromosomes, I should say, and their sex determination does follow the same homozygous female, heterozygous male pattern that humans have. T.H. Morgan was studying trait transmission in fruit flies the same way that Gregor Mendel was investigating trait transmission in peas by developing purebred or true breeding types of flies that always had, in this case, red eyes, and another type of true breeding flies that had white eyes. So he was following eye color. And what he did was cross these two, and of course, not strangely, all of the offspring, whether they were boys or girls, had red eyes, and this did not surprise him. And then he crossed those and ended up getting the same ratio that he expected, a 3-to-1 ratio of red eyes to white eyes. However, there was a gender skew. He didn't see that ratio conserved in both sexes. He saw all of the females had red eyes, and half of the males had red eyes, the other half had white eyes. How could Morgan explain this using good old Punnett squares? I mean, if it just followed regular patterns of inheritance, then it wouldn't matter what the F1 offspring were. Certainly, if the red eyes were dominant over white eyes, he would get all of that F1 generation, regardless of gender, having the red eyes, and then cross those F1s, doing the Punnett square through. He would get that 3-to-1 ratio, but he should get that regardless of the sex of the fly. So this clearly, something else is going on. To understand how red-eyedness in Drosophila shows this bias when it comes to sex, we have to look at the genetics of sex itself. Now, here I'm talking about biological sex, so I'm talking about which chromosomes you have. And in humans, I'm not talking about gender or gender roles. That's a whole nother ball of wax. Here we're just talking the biology. So in humans and most other mammals and in fruit flies, it's that last 2 X and Y chromosome and the combinations that are going to determine sex. So it's 2 X chromosomes, excuse me, that are homologous, you get a female. And this gives gene redundancy that you see in all of the autosomes that occur in pairs like the sex chromosomes do in females. If there's an X and a Y chromosome, so we can call this heterozygous for sex, then it develops as a male. There's no redundancy here because these are actually non-homologous. There are different alleles that are encoded on the X chromosome than are what are encoded on the Y chromosome. But we can do a Punnett square for chromosomes just like we can for genes, because we know that it's actually whole homologs that are segregating upon meiosis here. So the man is either going to produce sperm that carry the X chromosome or the Y chromosome. The female the woman is going to provide eggs that have either the X chromosome or the X chromosome. And when we do the Punnett square, you see that we get a 50-50 sex ratio from the chromosomes themselves. Now we can apply this segregation of sex chromosomes to Morgan's flies and sex linkage. So we'll say that the allele for red-eyedness is actually on the X chromosome. So that means that the female then is going to have 2 X chromosomes, both of which carry the dominant red-eyed allele. The male is only going to have 1 X chromosome, and he has the recessive white-eyed allele on his 1 X chromosome. Now, when we do the Punnett square, we go through and see that all of the females have red eyes, and all of the males have red eyes. And this explains how all of that F1 generation show the red eyes. When we take the females of the F1 who are heterozygous and cross those with the males of the F1 who are all just there, well, they can't be heterozygous or homozygous, they just have that one dominant red-eyed allele, and cross them, now we can see that what we end up with, let's cross all these guys out, are going to be all of the females having red eyes and half of the males having red eyes and the other half of the males having white eyes. If the sex chromosomes don't carry the genes for peepees and hoo-ha's, then how do they actually determine sex? Well, it really comes down to the Y chromosome, and it's just a little part of a chromosome, really. It doesn't have much in the way of genes on it at all, but it does have one very important section called the SRY, or sex-determining region of the Y chromosome. And its very presence, this one section of DNA, upregulates the production of male hormones and causes the development of male characteristics. So this one gene has lots of effects, which is a good example of pleiotropy like we talked about in the last video. The X chromosome, which is also a sex chromosome, has lots of other genes on it. It has as many genes as you would find in any other normal autosome. Mutations associated with the X chromosome and therefore inherited via the X chromosome and X-linked pattern of inheritance are things like hemophilia or muscular dystrophy, or more commonly, color blindness. To give you an idea of some of the mutations that can take place on the X chromosome and therefore cause problems, this also means that there must be genes at each one of these regions as well. There are quite a few problems or mutations that we refer to as sex-linked. And anytime we really talk about sex-linked, we're really talking about the X chromosome. It's a little weird since it's the Y chromosome that's really doing the determining of sex. But there are more than 60 diseases on this list that are associated with the X chromosome. Hemophilia is another sex-linked trait, and it does tend to run through some royal families. And this makes their pedigrees, the transmission of traits from generation to generation, rather interesting. And we're going to take some time in class to do some pedigree analysis. Before we can analyze an entire family tree for the transmission of a trait through several generations, we should know something about hemophilia and the way it is transmitted from just one generation to the next. Hemophilia can be a genetic disorder where an individual can't make a particular clotting factor. That clotting factor is coded for on the X chromosome. So if that is mutated and defective, it works as a sex-linked recessive trait. Now, let's see, we have a mom who is a carrier, so she is heterozygous. She's not a hemophiliac, but she carries an allele for hemophilia. Dad only has one X chromosome, and lucky for him, that one X chromosome is normal, carries a normal gene for hemophilia. Might have some other problem with it. We don't know. We're just looking at hemophilia here. So when mom makes gametes, she makes gametes that carry the X chromosome with the normal clotting allele, or gametes with the X chromosome with the abnormal hemophilia allele on it. Dad is going to make gametes that carry the X chromosome with the normal allele or the Y chromosome. Doesn't carry either allele for hemophilia at all. So when he makes sperm and mom makes eggs, and we go through the Punnett square, we see, lo and behold, that half of the offspring, half of the boys and half of the girls, aren't going to carry or have a recessive allele for hemophilia at all. In fact, none of the girls are going to phenotypically be hemophiliacs. However, half of them will be carriers. The boys, however, because they only have one chromosome, they don't have the opportunity to be heterozygous. That either have that allele or they don't have the allele. So half of them are going to have the hemophilia allele and therefore show the disease. So it's this lack of heterozygosity in males when it comes to X-linked traits that allows them, when it comes to X-linked recessive traits, have that trait more often than the females do. Now, in addition to this bias, this sex bias when it comes to phenotypic expression, X-linked traits have some interesting patterns of phenotypic expression in females. Because they have two X chromosomes where males only have one, in order to kind of balance out the males only having one of those X chromosomes in the female is going to be shut down into what's called a bar body. And this bar body does not ever get expressed from then on, from that embryonic cell on down the line. And it's random which one is shut down into a bar body and which one is expressed. If the individual is heterozygous about a trait on the X chromosome, it's going to lead to a mosaicism or a patchwork arrangement of that trait. That's because when the female whose body cells are heterozygous on that X chromosome, when one of them gets shut down, one of those X chromosomes gets shut down, in one cell, or one cells that come from that one cell, are only going to express the dominant black trait, where other cells are going to come from, groups of cells are going to come from one of those embryonic cells where the black dominant bearing X chromosome turned into the bar body, and only that orange one is expressed. And so you see these patches of orange, patches of black, and patches of white. That can only be a female that has that particular characteristic. Male pattern baldness sounds like it would be a sex-linked trait. It's a sex-influenced trait. It's autosomal. It's not on the sex chromosome. It's not on the X chromosome. There's an age aspect to this as well. It typically shows up after age 30. And it's kind of weird because it's a dominant trait amongst men and a recessive trait amongst females. So when we start looking at the genotypes, all a male needs is one dominant allele, and he's going to be bald. In order for a woman to be bald, but through male pattern baldness, she needs to have two recessive alleles. Now, we used to say that this is passed down through generations on the mom's side, and a man could tell if he was going to be bald by looking at his mother's father. And well, here's my mother's father. Here's Charles Card, and he is clearly bald. That's Duffy the dog, I believe. Now, my father, on the other side of the family, is also clearly bald. Heck, here's my brother, and he's bald too. Sorry, Sam. That's Sam, his son. He's going to be bald as well. In fact, here's my dad and my two brothers, and they're all bald. Now, to be fair, in this picture, my younger brother has shaved his head, but he's showing signs of male pattern baldness. Anyway, sorry, Jared. Me, on the other hand, though my grandfather Poppy was quite bald, I am not. This means that it's not just the genes that you have that determine your phenotype. A cell that has male pattern baldness genes, dominant male pattern baldness genes, will lead towards baldness if it's developed in an environment that has lots of testosterone, not a whole lot of estrogen. That same cell that's reared in a different environment with not a whole lot of testosterone, lots of estrogen, is not going to develop male pattern baldness. So it's not just the genes that you have, it's actually the interaction between the genes and the environment that really brings out the phenotype that we see. Now we see this in people of Northern European extraction when it comes to skin color. The genes for regulation of melanin, or for the making of melanin, are upregulated, I should say, when exposed to ultraviolet light. We can also see this in other organisms like the Arctic fox, whose coat color is influenced by heat-sensitive expression. As the temperature goes up, the production of melanin is also upregulated in the hair follicles. Hydrangeas are affected by the pH of the soil, so you can make your hydrangea more purple or more pink depending on the alkalinity. And I think we'll stop there for now. Again, thanks for checking into Mr. Ulrich's landofbiology.com. Feel free to ask me any questions by sending me an email or dropping them down in the comments section. Other than that, we'll see you in class.